morning, student. We will continue the topic protein metabolism, competition number 5.4, and today's subtopic is metabolic pathways of phenylalanine and tyrosine, their role in biosynthesis of various specialized biomolecules and associated metabolic disorders. Phenylalanine is essential amino acid, therefore it has to be supplied in the diet. Tyrosine is non-essential amino acid and it is synthesized from phenylalanine. Tyrosine is glucogenic as well as ketogenic amino acid. That means on catabolism, tyrosine forms glucose as well as ketone bodies. Tyrosine is also a precursor for various important compounds in the body like melanin, catecholamines like dopamine, norepinephrine, epinephrine, and thyroid hormones. So we'll see catabolism of phenylalanine and tyrosine. Or you can also see synthesis of tyrosine here. Step 1, the enzyme is phenylalanine hydroxylase. Here if you see on the top the structure of phenylalanine. Now in this phenylalanine, when OH group is added at para position, it forms tyrosine, which is shown on the right side. Therefore, tyrosine is also called as para-hydroxyphenylalanine. As the enzyme incorporate hydroxyl group in this reaction, the name of enzyme is hydroxylase. That is phenylalanine hydroxylase. Here oxygen is molecule is converted into water. First oxygen molecule is broken down. One oxygen atom is converted into water molecule and another oxygen atom is added to the tyrosine in the form of OH group. So basically this enzyme break the oxygen molecule into two atoms and add one oxygen atom in two different products, water and tyrosine. So such type of enzymes are also called as monooxygenase. Now in this reaction, tetrahydrobiopterin act as a coenzyme which donates two hydrogen atoms to the enzyme and get oxidized into dihydrobiopterin. This dihydrobiopterin is synthesized from biopterin. Now this oxidized dihydrobiopterin is reduced to tetrahydrobiopterin by enzyme NADPH dependent reductase. For this reaction, coenzyme is NADPH which supplies two hydrogen atoms. After formation of tyrosine, we will see catabolism of tyrosine. So this pathway shows catabolism of tyrosine. First, tyrosine undergoes transamination where amino group from tyrosine is removed and added to alpha ketoglutarate to form glutamate. This is a transamination reaction. The enzyme is tyrosine transaminase and coenzyme is as usual pyridoxal phosphate, which is essential for all transamination reaction. After transamination, tyrosine is called as phenyl hydroxyphenyl pyruvic acid. All of us know that another name of tyrosine is para-hydroxyphenylalanine. So after removal of amino group, that alanine group is converted to pyruvic acid. Now this para-hydroxyphenyl pyruvic acid undergoes one more hydroxylation with the help of enzyme para-hydroxyphenyl pyruvate hydroxylase. This enzyme again break the oxygen molecule into two oxygen atoms. One of the oxygen atom is added to the substrate in the form of OH group. So para-hydroxyphenyl pyruvic acid has now two OH groups, therefore it is called as dihydroxy, and at the same time the carbon dioxide is removed from pyruvic acid and acetic acid is formed. So here two products are dihydroxyphenyl acetic acid and carbon dioxide. The enzyme breaks the oxygen molecule into two atoms. One oxygen atom is added into the product dihydroxyphenyl acetic acid, and one oxygen atom is added to the carbon to form carbon dioxide. The another name of dihydroxyphenyl acetic acid is homogenesic acid, which undergoes oxidation. Homogenesic acid has a ring structure. That ring is opened by addition of oxygen molecule with the help of enzyme homogenesic acid to form a straight chain structure called as 4-mallyl acetoacetic acid, which requires coenzyme vitamin C. In the next step, 4-mallyl acetoacetic acid undergoes isomerization with the help of enzyme isomerase and coenzyme glutathione to form fumaryl acetoacetic acid. Now this fumaryl acetoacetic acid is broken down into two compounds by hydrolysis, enzyme hydrolase, and these two compounds are acetoacetic acid and fumaric acid. Acetoacetic acid is converted into ketone bodies and fumaric acid is later on converted into glucose. Therefore, in the beginning I have told you that tyrosine is both glucogenic and ketogenic amino acid. So this is about the catabolism of tyrosine. Now we will study the disorders of phenylalanine and tyrosine metabolism. Three main disorders are there: phenylketonuria, alkaptonuria, and hypertyrosinemias. Again, it is of three types: tyrosinemia type 1, which is also called as hepatorenal tyrosinemia; tyrosinemia type 2, also called as oculocutaneous tyrosinemia; and hereditary parahydroxyphenyl pyruvic acid deficiency. We will study one by one. Phenylketonuria. It is a very important disorder. Incidence 1 in 25,000 birth, autosomal recessive disease, and it is due to gene mutation. Again, coming to the first reaction, where we have seen phenylalanine is converted to tyrosine with the help of enzyme phenylalanine hydroxylase. So, if this reaction does not occurs, then phenylalanine is not converted into tyrosine, and phenylalanine get accumulated in the cell. Basically, this reaction requires primary enzyme phenylalanine hydroxylase and secondary enzyme which is required for regeneration of tetrahydrobiopterin is dihydrobiopterin reductase. Now, if you see the causes of phenylketonuria, the first and most important cause is deficiency of primary enzyme phenylalanine hydroxylase. Second cause is deficiency of secondary enzyme, that is dihydrobiopterin reductase, and the third cause is deficiency of enzyme synthesizing the biopterin. So I have told you that dihydrobiopterin is synthesized from biopterin, and that is converted into tetrahydrobiopterin, which is a coenzyme. So if this biopterin is not synthesized due to deficiency of enzymes, then this rate of this reaction decreases, which leads to accumulation of phenylalanine in the cells, leading to a disease called phenylketonuria. So these are the three causes of phenylketonuria. It is mentioned here. Depending upon the cause, there are 5 types of phenylketonuria. Type 1, also called as classical phenylketonuria, it is due to deficiency of enzyme phenylalanine and hydroxylase. Type 2 and type 3 phenylketonuria is due to deficiency of enzyme dihydrobiopterin reductase. So in both cases, the enzyme is deficient. In first case, the enzyme deficiency is more, whereas in second case, enzyme deficiency is less. Type 4 and type 5 is due to deficiency of enzyme synthesizing biopterin. Again, depending upon the severity, they are classified as type 4 and type 5. Now, when the phenylalanine is not converted to tyrosine, it gets accumulated in the cell. Then it goes in alternate pathways. Phenylalanine undergoes transamination. This pathway already exists in the cell, but when that pathway phenylalanine to tyrosine is existing, this pathway is minor pathway. Now due to blockage of that pathway, this transamination pathway is now become the major pathway in the cell, where phenylalanine donates amino group to alpha ketoglutarate to form glutamic acid. The enzyme transaminase or amino transferase, and phenylalanine is converted to phenyl pyruvate. Now this phenyl pyruvate undergoes reduction to form phenyl lactate with the help of enzyme reductase, where 2 hydrogen atoms are donated by NADH. Or this phenyl pyruvate undergoes decarboxylation reaction to form phenyl acetate. Now this phenyl acetate combines with glutamine to form phenyl acetyl glutamine. It is a conjugation type of reaction. So due to blockage of pathway which convert phenylalanine to tyrosine, so these are the alternate pathways and intermediates which get accumulated in the body. First is phenyl pyruvate, phenyl lactate, phenyl acetate, and phenyl acetyl glutamine. And due to accumulation of these substances, it will lead to a disease called phenylketonuria, which has following clinical features. First and most important is mental retardation. Here, due to accumulation of phenylalanine and its product in the brain, leads to damage to the brain of a small baby. As this disease present since birth, so brain development will take place up to 5 years of age. So during that process, these substances along with phenylalanine damage the brain and patient have mental retardation. Next symptoms is neurotoxicity. As I have told you that in the beginning, from tyrosine, various neurotransmitters like dopamine, epinephrine, norepinephrine are synthesized. Due to deficiency of this dopamine, there is neurotoxicity is seen. As tyrosine is absent, dopamine is deficient. Next is hypopigmentation of skin. As the tyrosine is not synthesized from phenylalanine, melanin is not synthesized. Hence, the skin becomes hypopigmented. And last is mousy odor. Mousy odor means a odor of dead mouse, and this mousy odor comes from the body of a patient suffering from this disease because presence of phenyl acetate or phenyl lactate in the sweat of these patients. Diagnosis of phenylketonuria. First is measurement of phenylalanine levels in the blood. Normally phenylalanine levels are 1 to 2 milligram per deciliter. Here it is written 1 milligram per deciliter. It is normally 1 to 2. But in case of phenylketonuria, phenylalanine level increases. Next is Guthrie test, which is positive. It depends on the growth of Bacillus subtilis, which are grown on a specialized media where inhibitors are present in that media. But when we add a blood of phenylketonuria patient to that media, that inhibition is overcome and this Bacillus subtilis grows on that media. When this growth is seen, we call it as a Guthrie test. If you add blood of normal person which has very less phenylalanine levels, bacteria will not grow and Guthrie test is negative in normal person. Next is ferric chloride test. It is not a specific test for diagnosis of phenylketonuria. It is a general test used for screening on large scale. When ferric chloride test is in urine is positive, then you cannot confirm the diagnosis of phenylketonuria. Now such patients are selected and then The above two tests were done on this patient to confirm the diagnosis of phenylketonuria. Above two tests are tedious and expensive, but ferric chloride test is cheap. Therefore, it is used for screening all the newborns for diagnosis of phenylketonuria, not diagnosis, for screening of phenylketonuria. Treatment of phenylketonuria. As the main culprit in this case is increased levels of phenylalanine and its metabolites. And phenylalanine is a essential amino acid which is not synthesized in the body. The only source is diet. Therefore, we have to give a diet low in phenylalanine. The examples of such diet is tapioca, cassava food. Now, this type of diet is continued for a patient up to 5 years of age, because during the first 5 years of age there is a brain development, and after that patient may shift to other diet, but phenylalanine rich food should be avoided. Again, when a phenylketonuria mother becomes pregnant, she should shift to the above diet, simply because to save the baby from high phenylalanine levels. Next treatment is gene therapy, which is under trial. So this type of cases are asked on this topic. After phenylketonuria, next disease is alkaptonuria. As the name suggests, excretion of alkaptone bodies in the urine is called as alkaptonuria. It is an autosomal recessive disease compatible with life. Compatible with life means patient can survive with this disease. Incompatible with life means patient cannot survive. The cause of this disease is deficiency of enzyme homogentisate oxidase. This is the tyrosine catabolism pathway which we have studied. Third step, the enzyme homogentisate oxidase is required, which convert homogentisic acid into 4-mallyl acetoacetic acid. So this step is blocked, due to which homogentisic acid is not converted and get accumulated in the cell. Now this homogentisic acid enters into the alternate pathway, where the enzyme polyphenol oxidase convert it into benzoquinone acetate. Now this benzoquinone acetate get polymerized to form alkaptone bodies. These alkaptone bodies may get deposited in various cartilages in the body, like vertebral disc, nose, pin of ear, and majority of them are excreted into the urine. These alkaptone bodies are colorless, but in the urine, when that urine is kept for a long period of time, these alkaptone bodies combines with the oxygen present in the atmosphere and turns black. Now diagnosis of alkaptonuria can be done by this. First is blackening of urine on standing. Just now I have told you, alkaptone bodies combines with atmospheric oxygen and turns black. Then next test is Benedict test positive, ferric chloride test positive, and if you take an X-ray, then it shows parodic appearance due to accumulation of alkaptone bodies in the nasal cartilage. And this type of cases are asked on this topic. Here the last question is explanation of arthritis in this disorder, because alkaptone bodies are deposited in various cartilages and they will damage the cartilage. So in our joint, cartilages are present. If these cartilages are damaged, it leads to arthritis. Remaining questions are there in the previous slides. Now the next third disease related to phenylalanine and tyrosine metabolism is hypertyrosinemias, and it is of 3 types. So this is tyrosine catabolism. I have written in different manner. This pathway is same. So here is the pathway of tyrosine catabolism. Tyrosinemia type 1 or hepatorenal tyrosinemia is due to deficiency of enzyme fumaryl acetoacetate hydrolase, which is responsible for conversion of fumaryl acetoacetate into fumarate and acetoacetate. Second is tyrosinemia type 2, also called as oculocutaneous tyrosinemia, due to deficiency of enzyme tyrosine transaminase, where I have indicated 2 there, due to which tyrosine does not undergo transamination to form para-hydroxyphenyl pyruvic acid. And third disease is para-hydroxyphenyl pyruvic acid hydroxylase deficiency at the second step of this pathway. So these are 3 types of hypertyrosinemias and their enzyme deficiency. In our pathway, these are the 3 enzymes and corresponding disorders. After studying the metabolic disorders, we will study the important compounds obtained from tyrosine. 3 important compounds I have told you: melanin, catecholamine and thyroid hormone. We will study one by one. First is synthesis of melanin. Tyrosine forms dihydroxyphenylalanine by addition of hydroxyl group to the tyrosine with the help of enzyme tyrosinase. Now these tyrosinase enzymes are present in melanocytes of the skin. This tyrosinase requires copper as a coenzyme or cofactor, you can say most properly. So in this case, again 2 hydrogen atoms are donated by NADPH and oxygen molecule is added. Now this tyrosinase is also a monooxygenase type of enzyme which break the oxygen molecule into 2 oxygen atom. 1 oxygen atom is added into the product in the form of hydroxyl group and other oxygen atom is added to the water. So here 2 products are dihydroxyphenylalanine and water. So for each product 1 oxygen atom is added. Therefore such enzymes are called as monooxygenase. Later on the same enzyme convert dihydroxyphenylalanine into dopaquinone. Dopaquinone spontaneously get converted to indolquinone and indolquinone get converted into melanin spontaneously. So in this way melanin is synthesized in the melanocytes which are present in the skin with the help of enzyme tyrosinase, and remember cofactor is copper. Now defect in melanin synthesis. As I have told you, copper is a cofactor for enzyme tyrosinase. In case of tyrosine deficiency, 2 types of tyrosine deficiencies are seen: absolute deficiency and intermittent deficiency. When there is absolute deficiency of copper is there, then melanin is not synthesized and such patients have whitening of hairs. If there is intermittent copper deficiency, means sometimes the person will get copper from diet or water and sometimes he will not get. At that time alternate dark or white hairs are seen. That means whenever copper is present, melanin is synthesized, the hairs are dark. Whenever there is copper deficiency, melanin is not synthesized and hairs becomes white. Next is leukoderma. Leukoderma are nothing but local white spots on the skin. They are due to deficiency of enzyme tyrosinase or melanocyte in some part of skin. That may happen due to damage to the skin at that position or damage to the skin locally or some part. Third disease is important, which is called as albinism, which is different from leukoderma. Leukoderma is local disease, whereas albinism is present all over the body. Here deficiency of enzyme tyrosinase completely, and and tyrosinase is absent since birth in the melanocyte. It is a autosomal recessive disease, incidence 1 in 20,000 birth. Here clinical feature includes the iris of the patient becomes grey or red due to deficiency of melanin in the iris. Normal iris is black in color because of presence of melanin. Next feature is photophobia. As iris guards the entry of sun rays inside the eye, and iris contain melanin. So due to deficiency of melanin, the iris cannot able to prevent the entry of sun rays into the eye. So in such persons, more sun rays enter into the eye, leading to photophobia. Nystagmus. The pupils of such patients are continuously oscillating, and there is decreased visual acuity. That means patient cannot able to see the object clearly. Due to deficiency of melanin in the skin becomes sensitive to ultraviolet rays, and hairs are white. Next defect in melanin synthesis is malignant melanoma. Here melanoblasts are also present in the skin. They are precursors of melanocytes. Here these melanocytes starts rapidly multiplying. Due to rapid multiplication of these melanocytes in the skin, the melanoblast contains melanogen pigment, which is a precursor of melanin. Now more and more melanogen is synthesized by the melanoblasts. This melanogen enter into the blood, filter through the kidney and excreted into the urine. Melanogen is a colorless compound. When this urine is kept in a test tube, it becomes black because melanogen get oxidized to melanin on exposure to air. So this type of case is asked on this topic. Next important product obtained from tyrosine is catecholamines. Catecholamines are the compounds which contains catechol nucleus. These catecholamines are synthesized either by adrenal medulla or sympathetic ganglia. Three types of catecholamines are important for our body: dopamine, norepinephrine, epinephrine. Epinephrine is also called as adrenaline. This is the pathway by which catecholamine is synthesized from tyrosine. The first step is conversion of tyrosine to dopa, just like melanin, but the enzyme is different. Here the enzyme is tyrosine hydroxylase, where this enzyme again it is a monooxygenase enzyme which break the oxygen molecule into two atoms. One atom of oxygen is added to tyrosine to form dihydroxyphenylalanine, means two hydroxyl groups, and another oxygen atom is added to water. So dopa and water, these are the two products which contain two oxygen atoms. So therefore it is a monooxygenase type of enzyme, and coenzyme is NADPH, which supply two hydrogen atoms in this reaction. Later on, this dopa or dihydroxyphenylalanine undergoes decarboxylation to form dopamine. Here the enzyme is decarboxylase. Now dopamine is the first neurotransmitter which is synthesized. Now this dopamine undergoes oxidation by combining with oxygen. Enzyme is oxidized to form norepinephrine. Here coenzyme like vitamin C and cofactor like copper ions are required for this reaction. Norepinephrine then combined with S-adenosyl methionine. Here non-epinephrine act as a methyl acceptor. If you remember transmethylation reactions which I have explained in the glycine metabolism. So this is a transmethylation reaction. First methionine is there, combines with ATP. Here ATP is hydrolyzed, 3 phosphate groups are removed, and adenosine group of ATP is added to the sulfur atom to form S-adenosyl methionine. Due to attachment of adenosyl group to the sulfur atom, the original bond between sulfur and methyl becomes weak. And that methyl can be easily removed and donated to any other compound. So in the next reaction, if you see, methyl acceptor is there, which accept the methyl and get converted to methylated product. Due to removal of methyl group from S-adenosyl methionine, SAM, S-adenosyl homocysteine is formed. So these are transmethylation reactions. Here norepinephrine act as a methyl acceptor, which accept a methyl group from S-adenosyl methionine and get converted into epinephrine. And after removal of methyl group, SAM is converted into S-adenosyl homocysteine. The enzyme is methyl transferase. Now actions of epinephrine. Epinephrine and norepinephrine increases the blood pressure by vasoconstriction. Epinephrine also increases the rate and force of myocardial contraction. Means it will act on heart. Epinephrine also relaxes the smooth muscles of bronchi. It causes relaxation of bronchi. Therefore, it can be used in asthma, where the bronchial tree undergoes bronchoconstriction. Epinephrine is anti-insulin in nature, which increases blood glucose level by glycogenolysis, means breakdown of glycogen. Also, it leads to stimulation of fat breakdown, that is lipolysis. Epinephrine is released from adrenal medulla in response to flight, fright, fight, exercise and hypoglycemia. Now catabolism of catecholamines includes the epinephrine undergoes one more transmethylation reaction. Here again epinephrine acts as a methyl acceptor, which accepts methyl group from S-adenosyl methionine, and that S-adenosyl methionine is converted to S-adenosyl homocysteine, and epinephrine is converted to metaepinephrine. The enzyme is catechol-O-methyl transferase. Later on, that metaepinephrine undergoes deamination reaction by enzyme monoamine oxidase. When ammonia group is removed in the form of amino group is removed in the form of ammonia, and metaepinephrine is converted to vanillyl mandelic acid. So the end product of catecholamine breakdown is VMA, vanillyl mandelic acid, which is excreted into the urine. When the synthesis of catecholamine in the body increases, excretion of VMA in the urine increases. So by estimating the VMA in the urine, you can diagnose the various diseases in which catecholamine synthesis increases. Now clinical importance of catecholamines. Dopamine is the inhibitor of prolactin secretion. Prolactin is milk secreting hormone. Dopamine is an important neurotransmitter in various tracts of the brain, that is extrapyramidal tract, substantia nigra, striatal tract. If there is dopamine deficiency in the body, then the functioning of this tract is hampered and the patients have various types of diseases. Drug alpha methyl dopa, which inhibits dopa decarboxylase enzyme, So second enzyme, dopa decarboxylase enzyme, which will convert dopa into dopamine, is given, which prevents the formation of catecholamines and also prevent the production of epinephrine. Therefore blood pressure becomes normal or blood pressure decreases. So therefore alpha methyl dopa act as a antihypertensive drug. Next, clinical importance of catecholamine is pheochromocytoma and neuroblastoma. So these are diseases. In pheochromocytoma, there is increased synthesis of epinephrine, and neuroblastoma there is increased synthesis of norepinephrine. So in both these diseases, the excretion of VMA is increased in the urine, because just now we have studied end product of catecholamine metabolism is VMA. So the diagnosis of these two diseases depends on estimation of VMA in the urine. Normally 2 to 6 milligram of VMA is excreted into the urine in 24 hours. But if the synthesis of epinephrine or epinephrine in above two diseases increases, then excretion of VMA is also increases. But when you are performing this test, VMA estimation in urine, then care should be taken that some compounds like banana, chocolate, coffee, citrus fruit, vanilla ice cream should not be consumed by the patient, because these compounds contains vanillin, and that vanillin in the body get converted into VMA and excreted through the urine. If you perform the test, that is estimation of VMA in the urine, after consuming these substances, naturally high VMA levels are obtained in the urine, so which may misdiagnose you. Therefore such compound should not be consumed by the patient. Now next important product obtained from tyrosine is thyroid hormones. Two types of thyroid hormones, T3, T4. T3 is also called as 3,5,3 triiodothyronine, and T4 are also called as 3,5,3,5 tetrahydrothyronine. Thyroglobin is present in the thyroid gland, which is a protein which contains tyrosine. When iodine atom is added to this tyrosine, it becomes monoiodotyrosine, MIT. If one more is added, it becomes diiodotyrosine. When this diiodotyrosine combines with another monoiodotyrosine, it forms triiodothyronine or T3 hormone. Diiodotyrosine, when it combines with another diiodotyrosine, it forms tetrahydrothyronine or thyroxine hormone. So in this way, two thyroid hormones are synthesized. Now diseases related to thyroid hormones: hypothyroidism and hyperthyroidism. Details of this hypothyroidism or hyperthyroidism will be studied during thyroid function test. Now hypothyroidism means decrease in thyroid hormones. It may be primary or secondary. Primary hypothyroidism means when thyroid gland will not able to synthesize T3, T4 hormone. And secondary hypothyroidism means when there is no stimulation by pituitary or hypothalamus to the thyroid gland due to defect in the pituitary or hypothalamus. There is decreased synthesis of thyroid hormones, which is called as secondary hypothyroidism. Hyperthyroidism means there is increase in the thyroid hormones. Again, it is of two types, primary and secondary. Primary hyperthyroidism means thyroid gland synthesizes more thyroid hormones without stimulation. That means defect in the thyroid gland. And secondary means when there is increased stimulation of thyroid gland by TSH, and more TSH is due to synthesis of more TRH. Then in such case either pituitary is defective or hypothalamus is defective. So they will synthesize more stimulating hormones. Thyroid is normal, but due to stimulation it is synthesizing the more thyroid gland. In such case it is called as secondary hyperthyroidism. So in both primary hypothyroidism and primary hyperthyroidism, thyroid gland is defective, whereas in secondary hypothyroidism and secondary hyperthyroidism, either pituitary or hypothalamus is defective. So this is the summary of tyrosine metabolism. Thank you very much.